Hey, Mike, just wanted to let you know that we are currently streaming out on YouTube, and I will, as usual, start the recording right at 2:59, and I'll send you a message once I've done that so that you'll be able to kick it off.
Sounds great. Thanks, Mike.
No problem.
Hello.
Hey, Pat.
Sorry, I lost the link.
Oh,
With
that's
two
okay.
emails, it's just a mess.
Oh, you have an aquatic theme. I'll put my aquatic theme on. How you doing?
Good. How are you?
Oh, quite well, actually. There, how's that? Huh.
Oh, nice. Where is that?
That's actually very close to my house in Arapika, Florida.
Oh,
We live on a salt marsh. I'll show you. You may have seen the salt marsh background, but this is. See if I can show you.
huh, you live in Arapika?
Well, much of the time. And I come up to Gainesville. It's just a short drive. It's an hour or so drive. I come up to Gainesville during the week when I'm teaching. Of course, now we're doing it all remotely. But that behind me is the outside my office window.
Nice.
It's pretty wonderful. And in the other direction is the Gulf of Mexico. We're right on the salt marsh, and wow. I can take no credit for it. My wife built the house 23 years ago. So, wow. Yeah, and she's a professional artist, and the entire third floor is her studio. So nice. And here I'll do this one. Here's another one. That's me and my boat coming home from. So anyway. Oh, nice. Yeah. Get all that water behind us. I sent a note around to Mark Brenner. I don't know if you know Mark. He's in the geology department here, another limnologist. But his colloquium, his seminar series, the same time as ours. And he runs his seminar series too. Oh. So, yeah. So he's not going to be able to join us, but that's too bad. Anyway. And maybe I should test to make sure I can share my screen. Yeah, give it a try. And if you can't, we need to get Mike to give you permission. Yeah, it says host disabled. Okay, Mike, if you could share, maybe I can do it. Let me see. Yeah, I can do it. Okay. Try that. This will stop other. I might as well try it. Let's see. Yeah. Yep. Do you see that? I see it. Got your Michigan State helmet there. Yeah. Hold on. Is that still viewable? Yep. That looks great. Oh, nice logo. Who designed that? We had a web person design it. With input from us. We were. Yeah. Is that an epilimnion, metalimnion, hypolimnion? No, actually it represents the three different major colors of lakes: blue, brown, and green. Yep. Yep. Okay. Yeah, we'll get started. We're already Zooming on YouTube, and Mike starts the recording in about a minute. Okay. And then at 3 o'clock on the dot, I'll make the introduction and then turn it over to you. Thank you very, very much for doing this too. This is sure. The one bright spot in all this COVID shutdown stuff is we can get people from all over to come give talks to the department. Without spending much. It's a lot easier to, give the talks. Yeah, yeah. Hello, everybody, and welcome to the Geography Department's 6th colloquium of the fall term. Along with the theme that we've been trying to follow this year about biologists doing geography and geographers doing biology, we bring you Patricia Soranno. Dr. Soranno is at the Department of Fisheries and Wildlife from Michigan State University. She's also a rotator division director at the National Science Foundation. And as I'm working part-time still for NSF, she's my boss. so it's great fun to have Pat here. Her bachelor's degrees from Notre Dame University and her master's and PhD are from University of Wisconsin. My advisor's PhD was University of Wisconsin, but many, many decades before. So it's a long, long legacy of limnology and limnological research and wonderful students coming out of the University of Wisconsin for a long time. Anyway, to get on with it, without further ado, I'm going to turn this over to Dr. Soranno, who will talk about the Lagos US database, leveraging open science, ecoinformatics, and geography to build research capacity for national scale environmental research. So Pat, have at.
Thanks, Mike, and thank you all very much for this invitation. As I was telling Mike before this started, it's great to have an opportunity to speak across the country and not have to travel really, and for myself to hear talks. So this is a great opportunity for me. So I'd like to start, I'm going to be talking about really our research program for the last 20 years, really. It's taken us a long time to build this capacity. And I want to start with answering the question of why. Why have I spent intensely the last 10 years on this effort, and more broadly, the last 20 years leading to this effort? And I'm going to answer why at the national scale a little bit later, but I also mean just in general at very broad spatial scales. So the reason we do this and why I think we need to do it more is because many environmental problems require it. And to me, I think the best example of why we need a macro scale or macro systems approach to studying natural systems comes from pathogen spread and disease-borne vectors. And I know that you have your faculty, Sadie Ryan's lab, and I believe one of our collaborators is speaking next week on this topic. This is not my area, but I would say if I had to go back to grad school again, this is what I would study, because it is a great example of how we really need to know fine scale information, but also the broader context of the region, the state, and beyond to the continent. And the spread of mosquitoes and the diseases that they spread is just a great example for that. But a little more closer to what I conduct my research in, we also need this national scale view for a lot of environmental policy. So for example, for setting water quality standards in lakes and streams. So if you're not familiar with water quality standards, they are mandated by the EPA to the states to determine state level standards. Now, even some states are quite large. For example, Florida has 10,000 lakes. It has the third highest number of lakes in the conterminous US. You may not know that statistic, but when we think about the lakes of the US, we're talking almost 500,000. And so how do we think about setting standards in lakes that are going to look very different? And here's just a couple examples. I grabbed one close to your home that I'm sure a lot of you have been to. But even if you're thinking about, say, just the state of Minnesota, you can see those two examples there, one very brown lake and one very green lake. And the question is, should they be applied? Should the same standard be applied to them? So to me, this is very much a macro scale question. So let me just dive into water quality just real briefly for those of you that don't spend your days and hours in life thinking about water quality. So water quality in lakes is most often measured by looking how green the lake is. And that green water comes from algae that grows throughout the open water zone. Algae, here's some charismatic algae that are very easy to identify, but the vast majority of algae are really hard to identify. So measuring them under the microscope is really pretty hard to do. So instead, what we do is we take a water sample, we measure chlorophyll concentration. We also can measure how clear the water is, how much light goes down. And we also can measure the phosphorus and nitrogen because there's very strong relationships, just like on land. If you add more fertilizer, you're going to get more green stuff growing in the water. So we can use these as really good surrogates to help us set standards. So all of these are measures of water quality, and each have important characteristics and information to help us better understand lakes. So what controls water quality? So lakes are very much embedded in their surrounding landscape. However, I would say, you know, through the history of limnology, the study of lakes and streams, a lot of people have focused on individual lakes. For example, for my PhD, I spent 4 very intensive years studying one lake. And it seems weird to say that now, but indeed I did. So individual lakes, we know from those studies that they're very connected to the surrounding land. They're also connected to other water bodies. They also are subject to the regional hydrology, such as runoff and groundwater. They also are influenced by the geology and the topography, which influences how the water runs off from the land to water. And of course, land use and atmospheric deposition are important drivers. And I know this last issue is a pretty big problem for Florida, in particular for the Everglades, as well as for all the 10,000 lakes you have in Florida. Now, for an individual lake, it's fairly straightforward to compile all these data and to understand a single lake. But how do we do that for 500,000 lakes? That's really the question. Now, as I alluded to before, across the history of environmental science, and I'm using that broadly to mean biology, ecology, and other sciences focused on natural communities at a range of scales. And I would say the majority have been studied at really the local scale. So in looking at a standard hierarchy here of natural systems, you can see there's 4 categories that I would put in the local scale, from ecosystem down to the individual. And then you jump from ecosystem to biosphere. Well, this is not something new, and I know Mike has spoken to you about macrosystems ecology already. And I would say for the last maybe 30 to 40 decades, ecologists have known that we need to sort of fill in this gap. And for example, so those studying landscape ecology, those studying ecoregions and biomes, and more recently, macrosystems ecology fits within this intermediate range. And that's certainly where I've spent the last couple of decades. Okay, so in thinking about all of this local scale knowledge, and we have a lot of it built up across the decades, what do we often do with this knowledge when we're trying to think about better understanding, say, local ecosystems? Well, one thing we do is we often extrapolate. So in this cartoon here, imagine these are lakes. Lakes are really good model systems because they have discrete boundaries. So you can count them and study them really easily. So think about 4 lakes, say in Florida, that we've sampled, and there's many more that we haven't sampled. What we can do is take these 4 lakes and we can extrapolate to the surrounding lakes, making the assumption, well, they're probably similar to the ones that we're studying. So that's done a lot with local scale knowledge. We also sometimes want to scale up. So we take however much data we have for lakes across some spatial extent, we scale it up to the continent, then we scale it up to the biosphere. Now, you're probably familiar with studies that do this, say, for forests. And we also do this for lakes and reservoirs to see how they contribute to global carbon cycles. And you can imagine we do this with very little data, although we probably have more data on forests than we do on lakes. And then the third thing we want to be able to do is forecast. So let's say these 4 lakes have pretty good historical data. We can then take that historical data, the current data, and then predict the future or forecast the future. I would argue most of the time when we're trying to understand environmental systems, we're doing one of these three things. And when we start entering the science policy interface, we really start doing this. So I would argue, is local scale knowledge sufficient to be able to extrapolate, scale up, and forecast? And so just to sort of think about that, let's do a little thought exercise here and think about what some of the challenges might be if we want to extrapolate, scale up, and forecast from a finite number of local systems. So I've taken the lake that I studied in Wisconsin, Lake Mendota here, and I've shown you, so I did my field work in 1992 and 93, and what you can see, what worked great for me is, so this shows phosphorus concentration in Lake Mendota. In 92, we had a pretty unusually low year for phosphorus, not the lowest, but a very low year. And then the next year was a pretty high year. It's great if you're doing a dissertation that's only 2 to 4 years, because you get a really nice contrast. So, okay, so let's say we've got this one lake. And if our goal is to forecast change in all lakes, then we start making assumptions about how lakes respond to this one well-studied lake. First, we can assume that lakes start out with different levels, say of phosphorus, but then they respond individualistically. And so here there's no relationship. One lake has a high, another lake has a low in the same year. In contrast, and imagine that we're talking about phosphorus here. In contrast, you can assume that lakes may start out different, but they're going to respond, say, to the same climate. So if it's a really wet year, you're going to get high phosphorus. In a really dry year, you're going to get low. And here we say that the temporal signature is synchronous in this case, whereas in the previous example, the temporal signature is ephemeral. So you can see when you're trying to forecast, you need to know, are you thinking, they're going to be synchronous, or are they going to be ephemeral? So we can also think about how ecosystems vary spatially when we're thinking of extrapolating and forecasting. So now the x-axis is space here, and we're comparing 3 regions. And let's say now we go out and sample 3 regions. We've divided the US into 3, and I've centered Florida as one of the regions here just for a frame of reference. And the question is, do lakes vary more regionally, or are they more individualistic, and do they vary more locally? So the first assumption we can make is that lakes within a region are more similar than lakes across a region. Pretty simple concept. And what this means is region 1 with Lake A, all the lakes around it are going to be more similar to it than the lakes in the other 2 regions. This is in contrast to the situation where we say lakes vary by local factors more than by regional factors. So this is lakes are responding, or lakes exhibit more individualistic behavior. And in one region, you could have really high nutrients and really low nutrients due to the very local specific characteristics. And so these would be lakes where there's more local spatial variation versus the first case where you have regional spatial variation. And so just to give you some examples, so we have data here. We have data for lakes where we've connected the mean temperature of the last 30 years to these lakes. And you can see a strong regional spatial structure here to these data, to mean temperature. And in contrast, a very weak spatial structure variable is lake depth. So this means if there's a shallow lake in one area, say Minnesota, there could be a deep lake right next to it. So this means there's no spatial structure, and so that's not going to help us. Lake depth is actually a really important variable to predict a lot of different factors about lakes. So the fact that it has no or very low spatial structure doesn't help us much. Okay, so what can we do with these type of data that I just described with these simple plots? Well, actually, we can do a lot with them. What we can do is break down the variance of lots and lots of data into these different spatial and temporal components. And what we can do is calculate the total percent variation of, say, total phosphorus across the landscape in all of these lakes as either regional, local, synchronous, or ephemeral. So this is a very simple box plot, bar chart, but actually to get that bar chart, you've got to use a Bayesian hierarchical variance components models with random effects. If you're interested in those details, you can look at some of our papers. It's a very effective tool because one thing it does is it accounts for very unbalanced sample designs, which these kind of data that I'm going to tell you about are very unbalanced. But it can factor and it can test for both local and regional predictors as well. So it's a really powerful approach. And so we've done some of this modeling, and I'm going to tell you the results of those. But I first want to tell you how we got here, because I wanted to make this plot, that variance component plot, probably 20 years ago. And I was not able to make this until just recently. We published these results in 2019. So it's taken a long time to get to that very simple plot. Okay, so what we argue, and Kendra, my longtime collaborator and co-director of my lab, we published a paper a couple years ago where we talked about this need to extrapolate, scale up, and forecast, and that quantifying spatial and temporal variance components really improves our ability to do all three of these things. Because if we know where the spatial structure is, it's really going to help us hone in on what kind of predictive models we need for all three of these activities that we want to do. Okay, but what does it take to be able to model spatial and temporal variance components? Well, we first need sufficient lakes sampled across all the regions. We need sufficient lakes sampled through time in different regions. We need integrated data sets. So most lakes are sampled at the state level or finer. So you have to combine all the data sets. You also need all of the great geospatial data sets that go with it to be able to build the model, to be able to make to test for whether local or regional predictors are more important for water quality. So currently, and I'm going to say this currently as of 2010, when we submitted our grant, and we actually submitted this grant to the Macro Systems program, and they agreed that it was a big gap, but there's no sampling program that covers all the US. There's no central repository for data, certainly that is research ready, and there's no associated geographic data that is all integrated to build the models. But what we thought and recognized is there's a lot of data hanging around in different parts that we could put together. Little did we know it would take 10 years plus to do that. Okay, so our proposition is that I'm going to spend the rest of the time talking about, that we'll be much better positioned to extrapolate, scale up, and forecast by explicitly studying the spatial and temporal scales between the ecosystems and the biosphere, i.e. the macro scale. And so what do we need to study these spatial scales? So first, we need the geospatial data that represent all of these scales. So not only just the broad scale, we also need the very fine scale. And to me, that's a really important tenet of macro systems ecology, is it recognizes the really valuable mechanistic fine scale knowledge that is gleaned from the local scale that can be applied to knowledge at the broader scales and vice versa. Second, we need research platforms and models to be able to pose questions and hypotheses at these scales. And then finally, we need concepts and theories for processes that operate at these broader regional to macro system scales. For example, can we just take the theories and concepts that are applied at local scales and just sort of scale them up? I would argue that we shouldn't do that. It's a starting point, but we should think about what kind of theory we need to study these systems at these scale. So as I said, it's taken us 10 years and we're still not done. But what we did is we first thought, well, let's start with the Northeast and Midwest region of the US, where there's mostly natural lakes, or we thought. That's turned out to not necessarily be the case. And where we know there's very lake-rich region and lots of good data sets. And so we have completed a database for this 17-state region. It's called Lagos Northeast, Lagos NE. And a lot of our results are using this database, because I said the national scale one's not ready yet. We wrote a paper in 2015 about how to do this, because as we were building this database, we realized there wasn't a lot of guidance in the literature about how to go about doing this. And in that paper, we argue for a very integrated approach of ecoinformatics, we called it GIS science, but basically geography, and ecology and other sciences. Now I've got statistics and machine learning here that I didn't put in the title today. It's also very important because to analyze the data, we've had to pull in very strong quantitative folks to help us with the challenges that we have with analyzing very large data sets, very unbalanced data sets, to make sure we're using robust approaches. Okay, as I said, the Lagos US effort is ongoing. So for this database, we had 51,000 lakes that we studied. We don't have in situ data available for all of those lakes, but that's our total census population. This database has, as I said, 500,000 lakes. So it's a bit of a jump up. Okay, so let's start with some of what we need in the geospatial data realm. So just to walk you through the different parts of the platform. So of course, our object of study are lakes. This part of the data set we call Limno, and we have lakes and we have sample sites, and they're collected at a sample depth, and then there's a sample event that's tied to a date. So there's about 200 data sets we're trying to integrate. And I would say there's 200 different ways to combine sample site, sample depth, sample event, and probably 200 different ways to call phosphorus. And I was going to make a figure of that, and I didn't get a chance to do it. But this was really a challenging part of this process. And it's taken, well, it took 5 years to do Lagos Northeast. It's taking another 4 years to do the US, and we're still not done. Then at the lake scale, we need to create a GIS base layer that allows us to geolocate those samples in a lake. And actually, that geolocating, geolinking step was perhaps one of our most challenging. And I would love to have a conversation about that, how we can improve that step. It's much harder than we thought. Then again, because lakes are very closely tied to their watersheds, knowing what a lake's watershed is critical. And there was no national scale watershed data set for lakes. So we had to create one. And that took roughly 1.5 years to do it for 500,000 lakes. And also, obviously, all of these methods have to be automated where possible. The methods with the Limno were where we actually had to do some manual processing because there was no other way to do it. And then finally, with the geo portion of the platform, we calculate with all the rich geographic data that's available at the national scale, calculate roughly 10,000 variables. And some of those variables are through time. They're at different zones, which means they're at the scale of the watershed, at the scale of a region, and we have multiple ways to measure regions. So we don't think all 10,000 of those variables are going to be valuable in any one model. It's to create the platform so future researchers can pick and choose. Okay, so the data is broken up into these 3 modules, Geo, Locus, and Limno. And the idea is that a module is tied to a certain type of data, and in particular, methods to summarize the data. And this modular nature of the data platform is a really important one. And I will say we did not have that idea from the beginning. As I said, some of this we've learned along the way. And it wasn't until towards the end of our first data product that we thought, you know what, these really are separate modules and we need to treat them separately, and it will benefit, you know, the use of the data set down the road to sort of have this modular nature. I think it was one of the better ideas we had across this whole effort. Okay, so why am I talking about the national scale? It's only because of the GIS data. So there is such a rich amount of GIS data available at the national scale to link to water bodies. And a lot of the data is much higher resolution and better quality than we can get at the global scale. So we did not take advantage of many global data sets just because of the rich national data sets we have at the US. Okay, so what's the platform besides just the database? Well, one aspect of the platform is that the different modules can be interconnected. And this is pretty obvious, but you've got to have unique identifiers that allow you to connect all of the data. You also have to have a data model that makes sense. Our data model is somewhat hierarchical, but it allows these connections. And then we provide additional resources. So for example, in our package to access the data, GIS base layers to allow future users to be able to, you know, create their own data, certainly to create their own maps. Extensive documentation. In fact, I've spent this entire year just writing documentation with my collaborators, and we're up to about 150 pages now. It's really a gruesome effort. But to make these data usable, we have to write the documentation. And there's two really key parts of this platform. One is we wanted it to be extensible. So that means sort of like Lego blocks, so that other researchers could create their own module and just connect it, snap it into the Lagos platform, take advantage of all the other data that we've got there. We've created a couple extension modules that have been sort of side projects on our own. So one is a data set where we differentiate between lakes and reservoirs. That's something that's also not available at the national scale. We've combined information on lake depth. Again, we did that by searching online for lake depths manually, and that's never been done. And then we've created lake networks. The second issue that we wanted is to make this data platform as research ready as possible, so that if you have a macrosystems ecology question about lakes, you can go to this platform and you can grab the data that you need to answer the questions that you need. So that's always been our driving factor. And importantly, the third important point is that the choice of which data to integrate and our data model were based on concepts and theories of lakes, landscape ecology, and macrosystems ecology. So even though we've spent a huge amount of time, you know, building a database that some may think, well, that's, you know, what kind of science questions are you doing? This is something that very much started with science questions and started with a conceptual model of how all these pieces fit together. Okay, so our principles, a couple of things I just want to highlight in building this platform beyond the ones I just mentioned is we thought it was really important to have a census population. So, and this is where the GIS data sets come in handy, because we can represent a lake on the landscape data without having data, so that people can reuse it. And we also have thought carefully about including data providers as co-authors. The remaining points are sort of standard open science principles, and I'd be happy to talk about those if you're interested. So just for a fun graphic, hopefully you're seeing this loop. So this shows from our first data product the time of sampling for water quality that's available from all these different data sets. So all of these data sets from these states, we went to the state agency and said, can you give us your data? And you can see that Minnesota wins the award for sampling their lakes for a long time. But then you see that in the 90s and 2000s is when the data really starts kicking up. But you see, we're not necessarily gaining new lakes after, the 2000s. So we are kind of continuing to sample the same lakes. So it's just fun to look at data like that. Okay, so what concepts and theories both informed the development of this and have we developed as a result of working on this project? Well, the first that I want to acknowledge is Landscapelimnology that we've been working on really since the 2000s, and in particular, along with Kendra, Kathy Webster, and Mary Tate Bremigan. And here we developed some ideas about thinking about lakes in a landscape context. And we borrowed heavily and shamelessly from landscape ecology. But what we realized at the time is that the majority of landscape ecology, when you look at the journal, was not written about aquatic ecosystems. And really, all of it applied: patch characteristics, patch context, patch connectivity, and spatial scale and hierarchy are really valuable for studying aquatic ecosystems. But up until this point, they hadn't really been studied in this way. And one thing that I would say differs from the terrestrial landscape is directionality really matters for water because water flows downstream. So there are some differences that you need to factor in for aquatics versus terrestrial. The second issue that we realized was really a concept that was important for aquatic systems is this idea of cross-scale interactions. And so this is where we recognize that there are local factors that really control how lakes respond to the landscape, but then there's also regional factors. And what we found is that the local and regional factors interacted. So the conceptual model on the left, imagine just a simple multivariate, well, a hierarchical multivariate model where the local variable and the regional variable are interactions. And what you get is, this means in the regional, so we have 35 regions, each region has its own slope when you're looking at the relationship between wetland cover and phosphorus. So lake phosphorus is our response variable. And so what you can see from these slopes is that if you combine all the regions together, there's no relationships. It's that really thin blue line. But if you look at a region-specific model, then there's some significant relationships that pop out. So then what you can do is you can model those slopes. You can say, what is predicting some regions to behave a certain way? And we found that the amount of agriculture in the region really influenced how local wetlands influence the lake. So this cross-scale interaction concept became really important. And we wrote a paper about it that sort of talked about cross-scale interactions a little more broadly, because we realized it was something that was really relevant to macro systems. We're not the first ones to study CSIs, obviously, but we helped to differentiate the different types of CSIs and the ones that were particularly important for macro systems, and the C and the D are obviously critical. Okay, and this around the same time, I participated in, I think this was the first macro systems PI meeting. We decided to do a special issue on macro systems ecology. And Jim Heffernan and I led an effort where we tried to define the term macro systems ecology. And I would say we spent 6 months just arguing about the definition. But I'm really proud of this paper and the special issue because we actually did reach a consensus. And it really got us starting to think about how we can think about these systems at broader scales. So we define it as the study of ecological phenomena at regional to continental scales, which sounds kind of simple, but it's doing ecology at these broader scales. So we came up with this framework, and I know you've seen this before, but what this framework shows is how local scales, the macro scale and the global scale might be connected, and the really important types of conceptual interactions that can happen. So you can see the pink there are the cross-scale interactions, then we've got the cross-scale emergences, and then macro scale feedbacks and the teleconnections. And we thought there were a lot of problems that could be placed in this framework. And it was just a really great experience. So anyway, so concepts and theories are really important. I feel like we're still in the early stages of that in relation to macro systems, and maybe Mike can give his thoughts on that in the Q&A session, but it's a really exciting time. Okay, so what about those variance components results with this data set that we've built so far? Remember, this is going into it, we expected about even spatial and temporal results or amounts. And so these are the data we had. We had data on about 8,000 lakes across 25 years. Not every lake had 25 years of data. It was very spotty, but it was from about 1990 to 2011. So you can see in the map on the left shows median TP in lakes. And obviously you can see the Northeast has lower phosphorus than say the agricultural Midwest. The plot on the right is the coefficient of variation. And this is just for graphical purposes. CV is not that useful of a way to analyze variance, but it's helpful to have a map. So as I said, this is what we thought we would see, and we were wrong. So we actually found much less temporal variation, very little of it synchronous. There's been a lot of literature about the synchronous nature of a lot of ecosystems, and so we expected to see it. And you can see also, we had a sizable residual, about 20%, where we weren't able to explain or assign the variation. So a couple caveats. We only included temporal variation that was monthly or longer. So that's a limitation of the data. We only modeled major nutrients and water clarity and chlorophyll, and we did not look at biological data. But what we also realize is that these kind of analyses really depend on spatial and temporal extents and resolutions. So these apply to this scale, but I would argue it doesn't apply to other. And I think we really need to do these kind of analyses at a range of different spatial and temporal scales. And so this is the scale we were able to study, and these results, I think, only apply to this scale. So what we can also do with these hierarchical Bayesian models is you can also then put in spatial, temporal, local, and regional predictor variables. So as you might imagine, we had a lot of spatial variation, so we could explain the regional and the local variation. These bubbles that are filled in means it's a significant variable. We accounted for covariance, there's no multi-collinearity in here. And the variables that popped out were ones that we would expect based on theory and knowledge of lakes. So it was really exciting to be able to compare these. And as you can imagine, we had so little temporal variation, we didn't have any significant predictors for any of the temporal components. So again, I'm a big fan of this approach. It's very powerful, and I think it's pretty fun to do. So what we found is that spatial variation is divided almost equally between local and regional scales for lakes at these macro scales. Ephemeral temporal variation far exceeds synchronous. But again, I think this is because we were looking across very large spatial extents. And past synchrony studies, I think were done at finer scales, So if you just hone in on, say, one region, you're probably going to find synchrony, but not across the extents we looked at. And spatial variation is 5 times temporal variation at these scales. So what are our opportunities for the future? Well, data volume and variety will continue to grow at all scales, at all ecosystem types, and also human data and social systems. Just increasingly, we're gonna have more data. So I would argue we have more opportunities to build research-ready platforms like the one I talked about today. Second is, I do think there's a critical role for the geographic sciences as environmental, ecological, and biological sciences broaden their spatial focus. And all of the data in the previous slide has a spatial and geographic component. So I think it's an exciting time to be a geographer. It's also an opportunity to address many more pressing societal challenges that I think are operating at these scales. But there are still real logistical and technical challenges in building research platforms necessary to do research at these scales. And again, the fact that it will have taken over 10 years to build a national scale system for lakes is, well, a little depressing, but I'm not sure, we're in much better shape than we were 10 years ago. So I will just leave you with that. I do think we mostly want to extrapolate, scale up, and forecast. And I think that thinking about both spatial and temporal variation and using methods like hierarchical Bayesian models are a good way to try to tackle this. And that's all. Happy to answer any questions.
Yeah, we have plenty of time for questions, Pat. And the person who usually asks the first question isn't here today, but I have a couple of questions for you, but I'll leave it for other people to ask first, and then I'll be able to ask get the last word in. Okay, actually, there's a question that comes in the chat. It says, what are the standards to define the selected lakes from other water bodies such as ponds when we're scaling up? Marv, maybe you should just ask the question. I'm not sure what you wrote here. What's a definition of a lake essentially? How do they differ from ponds? Do you include ponds? Or?
Well, yeah, so first I'll show you, okay. First, I'll show you some of the data we have from the national that's kind of relevant to this. So first of all, one of the things we have analyzed are lake names, and 83% of lakes are not named in the US, which is, we were shocked to find this. And the most common lake name is mud, followed by long. Early settlers were not that imaginative. So what we found really interesting is this graph. So this says what people call lakes. They call them lakes, they call them reservoirs, they call them tanks. That was new to us. And they also call them ponds. And so if you look at those red, not a huge amount of ponds in Florida. But what we found is that ponds have no ecological meaning. It's a cultural thing. And so like ponds in the Northeast are really quite large. So like in Maine, there's something called the Great Pond that is just huge. So in the Northeast, ponds are very large lakes. In the Southeast, ponds are generally smaller. But you're probably asking about small water bodies. And we use the cutoff of 1 hectare in our Lagos US data set. And that's just for a data quality reason, that the data of our base layer, the quality started to go down below 1 hectare. And really to define a lake, to me, a better term is wetland. There is the threshold between going from a lake to a wetland, and that has to do with depth, but we don't have much depth data. So to me, if you have a water body where you have, say, grasses growing all the way across, that's generally a wetland. So we try to have more functional definitions of lakes versus wetlands. versus open water all the way across.
Wow. Okay. Anybody else? That was a good question, Mom. But I have a question. A lot of the states have what are basically citizen science programs and have had for a long time. You know, and they've been collecting a lot of these data. Florida has the Florida Lake Watch. Minnesota's had a system for a long time. I assume you use those data in your database.
Oh yeah, Lake Watch is great. The Minnesota data set's great. It's just they do things completely different, so integrating them wasn't easy.
No, I understand that part, but you notice any variance in the quality of the data collected by the citizens versus collected by, say, state employees?
Well, I do not worry about that in the least, because in a lot of citizen science programs for water quality started up in the 80s and 90s. And so in the 90s, there were a lot of papers written comparing professionally sampled water quality with volunteers, and they found no difference in quality. And in fact, sometimes they found a little better with the citizens, because I think the professionals, you know, start getting a little sloppy. So to me, I have no concern about using volunteer data, and particularly at these scales, you know, it kind of gets lost in the wash. But I would argue we have greater sources of error and uncertainty than whether it's collected by a citizen, a volunteer or a professional. And I see someone asked a chat about the challenges or how we could set up a similar system in developing countries. And yeah, we've talked with some folks across the globe. And, you know, originally we wanted to do Lagos World. I'm so not doing that. But what we also realized is that I think something like Lagos needs to be at the national scale, because often sampling programs are at the national scale. So I think different countries should create their own. And then hopefully, you know, we would love it if people would use kind of the model and the system we've developed, and then it would be easy to collaborate and integrate the data. I think as far as other countries, it just depends on the resources they have available to them. We spoke with some folks, I believe it was Brazil, and they had an amazing sampling program. We just never got to the point where we took it to the next level.
I also had a question from a colleague who had to run, and I'll try to ask this in the way he meant it. He's concerned that there may be some kind of spatial autocorrelation between different variables that you're measuring. And one of the themes of geography is that close things are more closely related than far away things. Have you investigated any of the data set for spatial autocorrelation, positive or negative?
Yes. Yes, we've talked about this so much. So now I'm trying to think how best to. So the Bayesian hierarchical models help to some degree with that. We've also we've had some efforts where we've built models at the lake scale that model the spatial autocorrelation and take that into account. And yes, there is some spatial autocorrelation, but we don't believe it's impacting our conclusions about what's driving the water quality, because we're factoring in the fact that lakes to some degree are more similar near each other. That's not a very satisfying answer. We have talked about this extensively, and the interesting thing is just, I would point people to J.F. LaPierre's paper where what he did is he analyzed the spatial structure of all of the predictor variables and all of our response variables. And then basically the results show this interesting amalgam of spatial variation at regional and local scales in the nutrients, that the nutrients have both regional and local. It's just a really elegant study that he did, and that's the closest I can come. to that, but it is something we've thought about.
You know, I have a comment about Florida lakes. Most of the lakes in Florida are basically buried sinkholes, and where the sinkhole actually occurs way down below in the sand, below the sand mantle, and it puts a depression. You can find lakes that are 40,000 years old in Florida.
Yeah.
Like Annie and Lake Tulane and a few others. They can be within just a kilometer or two of a lake that was formed in 1985. But actually, I saw that happen. A sinkhole formed in Winter Park. It ate a Porsche dealership in a city park. And the lake's there now. It's a beautiful little lake. So, and it's not too far from Lake Tulane, which is 40,000 years old.
Yeah, and I mean, Florida, I'm really excited to add Florida to the database and start looking at it. That's a great point. And to me, that's a great argument for why we still need the local scale.
Yeah.
I would never argue or advocate that we can do everything at this very broad scale. You've got to look at the very detailed, local, and the regional. I would argue that I just feel like people have ignored the regional for a long time. So I'm just advocating we sort of add that to our mindset.
Yeah. All right, it's 3:50, so those of you who need to go can go for your 4 o'clock classes or whatever. And I'll stick around with Pat for another 10 minutes or so, and then we can, if you have any more questions, or maybe we'll just chat. Oh yeah, Mike Ryan reminds me to introduce next week's, in a second, it'll be Stephanie Mundus, who will be talking about, let me see if I can call it up here. Spatial patterns and mosquito characteristics that mediate vector control effectiveness and disease risk in Florida. Now, this is, Stephanie's a graduate student, and it will be her required colloquium. So students and faculty will have to make up write up the evaluations. So,
Sounds like a great topic.
All the grad students who are taking this for credit, don't forget to write up your evaluations and pass them in. And if Pat wants, I share them with her. Okay.
I did have a couple questions if you have the moment.
Sure.
I was wondering, you mentioned at the beginning about taking into consideration man-made versus natural lakes. Did you get the opportunity to, like, you said you didn't realize it till later, like you thought that there was a lot more natural lakes or something to that effect.
Right.
I know in West Virginia, where I'm from, there's very few natural lakes.
Right. Yeah, so what we found, so that's something, again, it's whether something has a dam on it and is an impoundment, and whether it was naturally formed, we think is really important to lakes. But again, there's been no way to classify what it is, again, for 500,000 lakes.
Yeah.
So what we did is we worked with a computer scientist to develop image recognition algorithm to classify whether it was likely to be a lake or a reservoir. And we, built manual models, and then we applied it to the whole 500,000 lakes across the US. So we now have a data product that says, you know what, this lake is likely to be a reservoir. Again, it's a prediction. And what we found is, and from our manual work, is that Minnesota has a lot of natural lakes, but it's got a lot of artificial lakes as well. And so in the upper Midwest and the Northeast, that's been glaciated, where there's a lot of natural lakes, you have a mix. Even though there's a lot of lakes in Minnesota, they still built dams. I don't know why, for a variety of purposes. Same with Michigan. So Michigan, we have a ton of natural lakes and a ton of reservoirs. But a place like West Virginia and Maryland, really a lot of things south of that, it's almost all reservoirs. So even with that, what I just said, when you actually look at the data, we really did need this computer science model to be able to tease it out.
I've been up to the UP of Michigan, up on with the University of Notre Dame Environmental Research Center, and all the kettle ponds up there are quite nice. My second question had to do with what package did you primarily use for the hierarchical Bayesian modeling? I'm just wondering. I use it somewhat frequently. Modeling tasks. Yeah.
Now I need to try to remember. We ran into problem between the two common ones. Oh, sorry. Ty Wagner did that modeling. Oh, we were so immersed in it a couple years ago. I'm sorry, it's escaped me.
No problem. I just wondered. I've started checking out INLA, integrated nested Laplace approximation, but MCMC-GLMM is one that I used previously. It just doesn't have, because of the that type of Bayesian modeling, it takes a long time for large models. So
yeah, so we're running into limitations like that as well. It's definitely cited in our 2019 paper. Sorry about that.
No problem.
It is not coming to mind. It was a while ago.
But thank you. Good talk.
Thanks, Sam. All right. Hey, so you could either you could calculate shoreline development index for each of these lakes if you've got a shape metric of some sort.
Oh yeah. No, we've calculated that. Yeah.
Wow. Is that a predictor of the nutrient? Is that a predictor of any of the nutrient concentrations?
It doesn't really pop out in the models. When you include, we've built a ton of these models to predict the nutrients. And I always thought it would, and it's not popped out to be that important.
Interesting.
Yeah.
That's always something we talk about in the ecology classes, is the extra contact that these long linear lakes have.
I know. And yeah, no, and actually we've calculated a few more shape characteristics, because I do think shape matters. And I do think the shape will help us better predict whether it's a natural lake or a reservoir, but we didn't have the shape metrics done to put into the model. But that's something that would be fun to do in the future.
Fun to do. Good master's project.
Yeah, exactly.
Okay. Well, thank you very, very much, Pat. I think we can start to log off now. There are only 6 people left, including you and me. Now 5. So, and it's delightful talk. Everybody seems to have loved it. They get a lot of good comments, and it's just the sheer magnitude of the work that you've done is overwhelming. It just blows me away. We took 2 years to map the forest management in our Macrosystems Award, and now we're working on the rest of the country, and it's going a lot slower than it did originally. We're finding the same thing.
Yeah. That this, even though the methods have been worked out, that it's taking us longer. I don't know why.
Part of our problem is that we did the Pacific Northwest and the southeastern U.S. first, places that members of the team had pretty good familiarity with the kind of forest management that's going on.
Right.
We have we're using a random forest classifier. It requires 1,000 basically training samples. And so we go in and we look in Google Earth at 1,000 spots. And in the southeast and Pacific Northwest, that was fairly straightforward. And we're trying to do the Midwest now. What do you know about forest management practices in the Midwest? Not much. We don't. We have people who are there.
Right.
Concord Desai at Wisconsin is one of our.
Oh yeah.
And he's helped us out a lot, but it's still. agonizing over a decision about what kind of management you're looking at. You know, we ran into Christmas tree farms of all things.
Yeah. Oh, wow. So how do you map the management? That's
We look at, excuse me, we look at the long-term phenology with MODIS data that goes back to 2000.
oh, okay.
16 days.
Yeah.
And that's one of the things, and a whole bunch of other ancillary variables that go in, the same kind of thing that you're doing with the lakes. And with a random forest classifier trained with all these things that now we still have to look at the training samples to train the random forest.
Yeah. Yeah.
And that becomes, again, it was straightforward in the places we knew that we did first, and it's now we're going farther afield. We actually have advisory committees in each of the regions who are trying to tell us what to look for.
Right.
And that's part of our program, and so they're helping, but it's just kind of clunking along now. So it's like
that makes me feel so much better, Mike, because I'm like, oh God, we're still not done.
Yeah. Well, we're in the 3rd year of our 5-year project. So
yeah.
I got kind of set back when I came to NSF, and I know that you're having, you don't have, about less time than the program officers do.
Yeah, so that's why all I've been doing since I came to NSF is writing documentation.
Yeah. And that's an evening weekends probably.
And it's just so freaking boring.
Yep.
But I mean, without the documentation, the data is useless.
Yep.
And it just takes so much. So I'm so excited to get back to science at some point, but until the data's out the door and it's documented, I can't do any science. So I'm very sad.
Oh, no. Yeah, you take a big bite.
Yeah.
Okay. Well, anyway, I should let you go, Pat. We need to get on to other things, and I need to
All right.
go down to Arapita this evening and go out and get on my boat this weekend. So.
Oh, how lovely. Well, enjoy.
Yeah. Well, you're in Michigan, right?
Yeah.
So anyway. Well, okay. Where is that behind your virtual background?
So this is a beautiful Japanese garden that was built on the western part of the state. A huge garden built around this lake. It's an artificial lake, but it's gorgeous.
Yeah, no, it's really quite nice.
Yeah, it's nice.
Anyway, so I'll see you again soon.
All right, thanks, Mike.